Hello, welcome to the PacBio ASHG 2020 CoLab. My name is Aaron Wanger, and my colleague Elizabeth Seng and I are both principal scientists at PacBio. Today we're going to talk about PacBio HiFi reads for comprehensive characterization of genomes and single-cell isoform expression. A HiFi read is a read that is both accurate and long. By definition, the reads are at least 99% accurate, or Q20 on the Phred scale, and typical accuracy is above Q30 or 99.9%. In terms of read length, HiFi reads typically range between 15 and 25,000 bases. HiFi sequencing is the best approach for the comprehensive characterization of genomes and transcriptomes. Key applications in human genomics are de novo assembly of human genomes, variant detection, full-length isoform sequencing. And key benefits are complete and accurate results, drastically reduced compute times and file sizes relative to other sequencing technologies, and lower coverage requirements. HiFi reads are available on the Sequel II system and the Sequel IIe system, which both are extremely reliable platforms that provide scale such that most projects can be achieved with one SMRT Cell 8M. The difference between the two systems is that with the Sequel IIe, which is newly released, you have direct access to HiFi reads directly off the instrument. This relieves the need to do additional computation of HiFi reads on your own compute hardware, which is how it works for the Sequel II system. PacBio has been adopted by key projects in human genomics. In the area of disease and cancer research, the Solve-RD consortium in Europe has adopted PacBio and is using it to sequence 500 individuals. And in population genetics, HiFi reads have been adopted by the Human Pangenome Reference Consortium, which is producing the next version of the human reference genome by combining sequences from 350 different human individuals. In terms of the two applications we'll discuss today, I'll first start with variant detection. And a variant is defined as a difference between an individual and the reference genome, and they range in size from small to large. On the small side are single nucleotide variants, or SNVs, which are base differences between the reference genome and an individual. For example, as shown here, the reference genome would have a C and the individual has a G. Then there are small insertion and deletion, or indel variants, which are typically defined as ranging in size from 1 to 49 bases. And this is where the individual either lacks or has extra bases at a position compared to the reference. And then finally, on the large side are structural variants, which are defined as differences that involve 50 or more bases. These come in a variety of different types, including large insertions and deletions, but also inversions, translocations, and duplications. PacBio HiFi reads provide a comprehensive view of variation in the human genome, spanning all classes from small to large. There are two main areas in which HiFi reads add variation that's not well detected with short reads. The first is in the area of long insertion and deletion variants, genome-wide. That includes examples like shown here. This is a 1,733 base pair deletion that's clearly delineated as a homozygous event in the HiFi reads. On the bottom are short reads for the same individual at the same position. And you can see these, the HiFi reads and short reads are concordant at the level of single nucleotide variants. But in the area of the deletion, there's ambiguous mapping of short reads because this deleted region lies in a repetitive sequence, and it's difficult to know whether the short reads that map here originate from this region of the genome or originate from another copy of the repeat elsewhere. Thus, it's difficult to detect this deletion with short reads. In terms of software for detecting structural variants with PacBio reads, we provide a solution called PBSV. It starts with aligning HiFi reads to the reference genome using a aligner called PBMM2. And then variants are called using PBSV in a two-stage process where first structural variant signatures are discovered, and then variants are called to produce a VCF file. And this workflow is available both at the command line with tools downloadable from Bioconda, and also integrated into the PacBio graphical user interface SmartLink suite. HiFi reads have been applied in rare disease to detect pathogenic structural variants that had been missed with short read sequencing. In this example, there's a preprint from earlier this year from Susan Hyatt and colleagues from Hudson Alpha Institute. And they applied PacBio HiFi sequencing to look at 6 individuals with neurodevelopmental disorders who had not been able to be diagnosed with short-read whole genome sequencing. For 2 of these 6 individuals, they identified a strong candidate pathogenic variant. One of them is shown here. This is their proband number 6, in which they detected a de novo duplication of exon 6 of the gene CDKL5 that was induced by a line element. Moving beyond structural variants, the second area where PacBio HiFi reads add variation that's not well detected with short reads is in difficult to map regions. One example shown here, this is the gene STRC. It's a congenital deafness gene that requires long reads to cover all the exons. So again, showing our reads from the same individual on the top short reads and on the bottom HiFi reads. And there's an area on the right-hand side of the gene, including many of the exons of STRC, that are not covered by short reads. And that's because there's a pseudogenized version of this gene that exists elsewhere in the genome that induces ambiguity in mapping. But HiFi reads are able to span from the unique sequence on the flanks through this duplicated sequence and delineate the single nucleotide variants, including many exonic variants that are missed with short reads. STRC is only one of a number of medically relevant genes that are not completely covered by short reads. This is one list from a paper describing what they call NGS blind spots. They note 193 known medically relevant genes that are not completely covered with NGS reads, and the vast majority of these are fully accessible with HiFi reads. This is also only one list, and there are likely more such genes to be discovered as more genomes are sequenced with HiFi reads. In terms of software for calling single nucleotide variants and indels with HiFi reads, we recommend the DEEPVARIANT variant caller from Google, which is a highly accurate small variant caller for HiFi reads. It's based on a deep learning model and is thus able to take example HiFi reads from control data sets, learn the properties of those reads, and then generalize to be able to call variants in other genomes. Now, the training of DEEPVARIANT and in general, rigorous measurement of performance of variant calling relies on benchmarks, the most common of which are available from or provided by Genome in a Bottle. Genome in a Bottle is a consortium hosted by the US National Institute of Science and Technology, and they provide benchmarks for 7 different human genomes numbered HG001 through HG007. Genome in a Bottle provides both physical DNA for those samples and a benchmark that shows regions of the genome where they're confident in the sequence and the variants that exist within those regions. This allows you then to take a call set from a technology like HiFi reads, compare to the benchmark, and identify true positives, false negatives, and false positives. The benchmarks that have been used for the last few years were released in 2017. They were the Genome in a Bottle version 3.3.2 benchmarks. These were built primarily using short read and linked read sequencing. They cover 85% of the human genome, but only 24% of the most difficult to map regions and segmental duplications and other low mapability sequences. At the end of 2019 and continuing into 2020, the Genome in a Bottle Consortium worked to extend these benchmarks using HiFi reads and other technologies, and they produced an expanded version of the benchmark for the sample HG002 that now covers 92% of the genome, including 54% of the difficult to map regions. They then proceeded to produce similar benchmarks for other samples, HG3 and 4, but rather than immediately releasing those benchmarks, they instead launched a contest called the PrecisionFDA Truth Challenge V2. Here they provided DNA sequencing data from the Illumina NovaSeq, the PacBio Sequel II system, and the Oxford Nanopore PromethION for HG2, 3, and 4. They provided the extended benchmark for HG2, but held back as blinded the expanded benchmarks for 3 and 4. Groups were then invited to submit call sets for HG003 and HG004, and the Genome in a Bottle group was able to compare the submissions to the not released truth set and score how well everyone did. There were a total of 64 entries to this contest. 24 of those entries used only Illumina reads. 20 used multiple technologies. All of those 20 included HiFi reads in their set. There were 17 submissions that used only HiFi reads, and there were 3 submissions that used only Oxford Nanopore reads. When the results were released in August of this year, the top 12 entries and 25 of the top 26 entries used PacBio HiFi reads. Here the entries are sorted from left to right, from best performance to worst performance, and colored by the technology that was used. The best single technology entry was from HiFi reads using the DEEPVARIANT variant caller. This performed significantly better than the same variant caller on Illumina reads, and dramatically better than DEEPVARIANT on Oxford Nanopore reads. It also outperformed Illumina reads with GATK, which is, of course, a very common workflow. Looking at it in terms of number of errors rather than precision and recall, PacBio HiFi reads with the DEEPVARIANT variant caller had 13,067 total errors in the 3 billion base pair human genome. The Illumina DEEPVARIANT call set had 2.5 times as many errors, mostly single nucleotide variant false negatives in difficult to map regions and false positives. GATK had 5.8 times as many errors, and the Oxford Nanopore Pepper DEEPVARIANT call set had 30 times as many errors, most of which were false negative and false positive indel calls. Looking at all variant types together, starting on the left with the single nucleotide variants to structural variants on the right, and here the number of errors are normalized to the PacBio call set. So you see in terms of single nucleotide variants, PacBio outperforms Illumina and Oxford Nanopore. In terms of indels, both Illumina and PacBio perform well, and Oxford Nanopore performs poorly. And as structural variants, both PacBio and Oxford Nanopore perform well, but Illumina performs poorly. So PacBio is the only technology that provides a high precision and recall, good performance across all 3 variant categories. In summary, PacBio HiFi reads provide the most comprehensive view of human genomes with high precision and recall for all classes of variation from small to large. PacBio HiFi call sets achieve the highest accuracy in the precisionFDA Truth Challenge V2. We have some more information on this challenge on the PacBio blog. In terms of variant callers, both PBSV and DEEPVARIANT are excellent variant callers for HiFi reads that respectively provide structural variants and small variants. And we recommend around 15-fold coverage as a good balance between cost of sequencing and accuracy of the results. So with that, I'll transition to my colleague Liz, who will talk about full-length isoform sequencing.
Hi everyone, my name is Liz and I'm a principal scientist at PacBio. My talk is on single-cell RNA sequencing using the Iso-Seq method. Traditional RNA-seq is done by fragmenting cDNA, sequencing it with short reads, then assembling it computationally. This transcript assembly method is unable to unambiguously identify full-length transcripts. In contrast, the PacBio Iso-Seq method sequences full-length cDNA. No assembly is required. Single-cell RNA-seq is used to study RNA expression at the single-cell level. Using short reads, however, captures only the 3' end of the transcripts, effectively limiting studies at the gene level. Using PacBio, one can study full-length isoforms at the single-cell level. HiFi reads are accurate enough to call both UMIs and single-cell barcodes with no error correction. In this pilot study, matching long and short read single-cell barcodes are concordant in their ranked abundances. Further, the sequenced transcripts are full length, ranging from 100 bases to up to 10 kilobases long. Theoretically, any single-cell platform that generates full-length cDNA can be sequenced on the PacBio systems. The precise locations of the UMIs and barcodes do not matter. Single-cell Iso-Seq has been done on a variety of platforms for different research areas. Below, I will highlight a few of these studies. In this preprint, researchers sequenced the mouse hippocampus and prefrontal cortex samples. Both short and long read data were generated. Using the short reads, they divided the hippocampus and prefrontal cortex into distinct cell types. Pairing the cell type information with long read data, they found 395 genes that showed differential isoform expression. 76 of those 396 genes were novel, as in not annotated in GenCode. One particular gene of interest is shown here on the lower right-hand side. This gene, NSF1C, is shown to have a 6-base pair microexon that is preferentially included in the hippocampus across the neuronal and glial cell types, but is absent in the same prefrontal cortex cell types. In another study, researchers sequenced cerebral organoids of human and chimp. As you can see here in gold and purple colors, between the chimp and human cells, there is a difference in the preference of transcription start sites. In this preprint, the authors used single cell to study expression changes in hematopoietic stem and progenitor cells in young versus aging mice. They were not only able to see certain isoforms being enriched in certain subclusters, they also saw that for the immunoglobulin gene IgK, the aging process led to an increased recombination event. In this final preprint, dubbed the Iso-Seq method, the authors aimed to increase the throughput of usable full-length reads on the 10x platform for PacBio sequencing. They used probe enrichment to remove TSO artifacts and concatenated the cDNAs to increase throughput by 8 to 10-fold. Here's the bioinformatics workflow for single-cell Iso-Seq. We begin with HiFi reads. Then cDNA primers, UMIs, and cell barcodes are extracted and removed. We then cluster reads that belong to the same founder molecules together and call a consensus. Founder molecules are determined by sharing the same UMI, cell barcode, and transcript sequence. Now that we have transcript sequences, we can map them to the genome and use the SQANTI3 tool to classify them against a reference annotation. SQANTI3 is a tool developed by the CONESA lab to classify long transcripts against a reference annotation such as GenCode, as well as use orthogonal data such as CagePeak, RNA-seq data to validate and filter library artifacts. Briefly, this is the nomenclature for transcript classification. Given a reference, a transcript is a full splice match if it matches the reference perfectly by the number of exons and the precise junctions. If the junctions and exons that are shared do match, but one or more of the exons are missing, it is an incomplete splice match. There are two kinds of novel isoforms. Novel and catalog are novel isoforms using a combination of only known junctions. Novel not in catalog are novel isoforms that uses at least one novel donor or acceptor site. The SQANTI filtering step aims to reduce the number of cDNA library artifacts. Most of these filter artifacts are coming from intrapriming or template switching artifacts. These transcripts that are filtered tend to be genic, genomic, antisense, or intergenic, and they tend to be monoexonic. After the SQANTI step, we can combine the cell barcode information to create a cell matrix that we see below. This bioinformatics workflow is now available in GitHub. To summarize, the single-cell Iso-Seq method is compatible with any full-length single-cell platform. It follows the standard Iso-Seq library preparation and sequencing workflow. We recommend generating matching short-read data from the same library. With a single SMRT Cell 8M, we expect generating up to 3 million HiFi reads. If you are interested in learning more about variant detection and the Iso-Seq method, here are some resources. Thank you for listening to our collab. You can come visit us at the collab networking table Wednesday, October 28th, noon to 1 p.m. Eastern Time. Or come visit us virtually at the PacBio booth anytime during ASSG. Thank you.